Vitamin A, roles of retinoids in type 2 diabetes. In this video, we are going to talk about effect on the pancreatic beta cell biology and insulin secretion. Retinoids are various synthetic or naturally occurring substances. Retinoids are made from vitamin A. Retinoids are one of the most widely researched ingredients for caring for maturing skin. Given this, it's no surprise that this class of vitamin A derivatives is often touted as the gold standard for reducing the appearance of fine lines, wrinkles, large pores, and more. Also, the actions of retinoids in liver fat accumulation. Scientists found evidence that vitamin A plays a role in how the pancreas makes insulin. Insulin is the hormone which regulates the blood sugar level inside the body and controls diabetes. Early research showed that certain proteins related to vitamin A are present in the pancreas, suggesting it's important for islet function. Studies in rats showed that when they don't get enough vitamin A, their insulin release gets messed up. This can be fixed by giving them more vitamin A. Vitamin A deficient rats displayed reduced insulin release when glucose is present, but this was reversed with treatment of the rats with a form of vitamin A before islet isolation. Insulin release remained reduced upon feeding with low levels of dietary vitamin A around 2 micrograms per gram diet, but were partially normalized at higher levels 8 micrograms per gram diet. Rats without enough vitamin A also have higher blood sugar and are less able to handle glucose, but this can be improved with more vitamin A. Interestingly, the pancreas of healthy rats has more of a specific protein related to vitamin A compared to rats without enough vitamin A. This suggests a connection between this protein and insulin release. Vitamin A seems to protect the insulin-producing cells in the pancreas from damage, helping to prevent diabetes. Scientists studied cells in the lab to understand how vitamin A affects insulin production. They found that vitamin A increased certain proteins, CRBPI and CRABPI, and also helped the cells release more insulin. This suggests vitamin A plays a key role in insulin production. Another study showed that vitamin A not only boosted insulin release, but also increased the amount of insulin instructions, proinsulin mRNA, and the machinery for using sugar, glucokinase. This supports the idea that vitamin A directly affects how cells make insulin. Scientists also looked at special receptors in the cells that respond to vitamin A. They found different types of these receptors in different animals, and that one type, RAR-alpha, is especially important in humans and rats. They even identified a part of the insulin gene that listens to vitamin A signals, further strengthening the link. Other studies confirm that vitamin A is crucial for healthy insulin production by helping cells make the tools they need, like sugar gates, GLUT2, and the sugar reading machine, glucokinase. Overall, the evidence strongly suggests that vitamin A plays a vital role in how the pancreas makes insulin. However, scientists are still working to fully understand exactly how this happens. Retinoid regulation of liver fat metabolism. There is accumulating evidence in the literature suggesting a role for retinoids in the regulation of lipid metabolism in the liver. While we are in the middle of the video, do not forget to hit the like button while watching this video. See the description and comments for references. We would appreciate your comments. Data from several human studies strongly support this conclusion. For a cross-sectional study of 91 children, a trend was observed between low serum retinol levels and an ultrasound-determined risk of liver accumulation of fat in tissues. For a case control study involving 138 adults, low dietary intake of retinoids, a form of vitamin A, was one of only 2 differing features between control subjects and those with non-alcoholic fatty liver disease. Another study of 145 obese adults reported that subjects with non-alcoholic fatty liver had significantly lower serum retinol and beta-carotene levels than healthy subjects. These human population studies suggest that lack of dietary vitamin A, beta-carotene, retinoid and carotenoid intake and the development of fatty liver. Collectively, these studies suggest that retinoids may be important and involved in the regulation of liver fat metabolism. Inconsistencies reported by different groups may arise from differences in study designs. Increased circulating free fatty acid levels may be responsible for increases in liver uptake of fatty acids. No clear conclusions regarding the effects of retinoids on liver fat metabolism can be established at this point. Your health is very important for your family as well. Do not forget to click the like button. Thanks very much for watching this video. Up above you see 2 videos related to health. It will be more helpful to you and your family. See you in the next video.